I have, so I'll now introduce our speaker. We have Wendy Chung with us here today. Wendy Chung oversees all aspects of Spark. She's also the lead of the Missense Variation in Individuals project, a study that aims to understand genetic differences that play a role in autism. She currently directs the clinical genetics program at Columbia University, where she performs human genetics research. She also assesses children with autism and other intellectual disabilities using advanced sequencing techniques to identify the genetic basis underlying neurological conditions. It's our pleasure to have Wendy with us today, so without further ado, take it away.
Thanks, Vincent. I'm really pleased to be here today. Today we're going to be talking about genetics and autism, and it'll be an update of a previous webinar I've given you based on new information that we have that's come directly from your research study from Spark. As we think about the genetic causes of autism, I want to emphasize that autism is not one single condition. It really is an umbrella that we use as a term to describe many individuals who share many of the core features. These are challenges in terms of social interactions, perhaps some limited scope of interest and repetitive behaviors. But they can come with different other challenges as well. For certain individuals, that really is all that they need to cope with, but other individuals may have other medical problems, for instance, epilepsy, sometimes gastrointestinal issues. Other individuals have particular behavioral issues. Some can have anxiety disorders, problems with attention, problems with deficits, or rather depression or attention. And for some individuals, this can actually change over their life course. There may be a certain set of things that are challenges early on and a different set of things that change either in adolescence or into adulthood. For that reason, we actually envisioned that Spark was going to be incredibly important for the community because we wouldn't have one answer or one size that fits all for everyone. And as we started thinking about Spark and about creating a community, we needed to have a large enough community that we could then take slices of the community that would have similar challenges. So for instance, we could take everyone who had autism and seizures and be able to think about their specific challenges. Other individuals who might be what we often call twice exceptional or have particularly strong areas that they're very good at, and be able to think about some of their challenges as well as their needs. By doing so, we might be able to understand better how the brain was working, what were the differences, as well as the rest of the body, and potentially be able to power or have sufficient numbers that we could see consistent patterns within those subgroups that might be more similar. We could be able to take out some of the heterogeneity or differences and be able to have large enough subtypes or subgroups that would be reproducible in terms of the research that we would see. And so that was one of the reasons for starting Spark and thinking about the very large scale that we've thought about doing it. So one of the components, and I want to emphasize it's just one of the components, but one of the components we focused on has been genetics. The reason why we started thinking about genetics was in part because we know based on some epidemiological studies, that there is evidence that genetics can play an important role in autism for some individuals. So let me just briefly review that that's because we can look in individual families and see sometimes that this aggregates, that we can have siblings or we can have parents or cousins or aunts or uncles, but that we can see this running in families. And so on average, we can see that if one sibling has autism, there's about a 20% chance that another sibling will have autism. There are particular special types of siblings, though, that of twins. And twins are special because they share the same intrauterine environment. Obviously, moms are pregnant with them at the same time. And there's one set of twins, so-called identical twins, that in fact genetically are, if not completely identical, almost completely identical. And so it allows us to, in a very special way, control for the prenatal and to a certain extent the postnatal exposures of those twins and make comparisons. And when we do so, I think there are two important things that we see. Number one is that within identical twins, if one twin has autism, there's a very high likelihood that the other twin has autism. In fact, that number is about 75%. But importantly, it's not 100%. So in other words, to me, that says it's not all in the genes. There are other things in addition to the genes that are important. But importantly, a portion of that is in the genes because For fraternal twins, or basically brother-sister or brother-brother-sister-sister twins, that rate of concordance, or that similarity in autism diagnosis, is only 31%. So distinctly different from fraternal twins and identical twins. The other reason for focusing on the genetics is because we really need to be able to understand at a very fundamental how the brain is working, how the body is working differently in individuals with autism, to think about how to come up with better supports for individuals. And as I said, it's complicated. There may be many, many different ways in which individuals both manifest and are susceptible to autism, and we needed to understand as much of that as possible. And so the genetics is a very robust way of being able to do that. We can be quite certain of the results when we see them, and so that seems like a good way in terms of going forward to identify at least some of the things that we could see in terms of risk factors. As we're doing so, one of the things we've learned in terms of genetics that is sometimes confusing is that there are genetic factors that are inherited. So these are things that are passed down from mothers and fathers. There are also things in our DNA or in our genetic makeup that start new with us, so-called de novo, or starting new. Both of those are encoded within the DNA, but only the inherited factors are passed down from generation to generation. And I'm going to go into more of this in detail. Let me also say, though, that genes interact with our surroundings, our environment, our things that happen to us prenatally before we're born, as well as those things, especially for autism, that are within the first few years of life as the brain is still developing and growing. We don't know all of those factors, and in fact, those factors are hard to pin down sometimes. But we do know that there are certain things that seem to increase the risk. They don't necessarily cause it for everyone who's exposed, but they can, in large populations, increase the risk. Some of those factors we think have to do with certain infections that, or exposures of viruses that the developing fetus may be exposed to while a mother is pregnant. Some of those same risk factors or infectious diseases may also be true after the baby's born, but still while the brain is growing and developing. There could be certain medications. For instance, there's a medication that's used for epilepsy called valproic acid, which has been associated with autism. We also know that prematurity, or being born too early before the brain is fully baked, if you will, that can increase the risk or risk of injury for an individual at risk for autism. So it's complicated. In any one person, it can even be more complicated in that there may not be one single driving factor. There could be a combination of multiple factors, but the point of Spark is to be able to learn as much about all of these dimensions, both the genetic as well as the environmental exposures as well. So I'm going to spend the rest of the time delving into the genetics, not to say that the rest of it isn't important, but we're trying to cover multiple topics in multiple different webinars, and today we're going to focus more on the genetics. So to give you a sense of how complicated things are, each one of us has approximately 20,000 genes that we have within our genetic information. With that, each of us has literally thousands and thousands of genetic variants. And that's what makes us all unique. And in fact, for each of us, approximately 1 in 1,000 of those spots is different from you and from me. Our genetic information is filled with 3 billion letters that make up that genetic alphabet. And so, as you can imagine, there are just many, many different places in which we're different. And the challenge that we have in Spark is to be able to see the difference between what are just differences that are quite common, or differences that might make up, for instance, why some people have brown eyes and some people have blue eyes, and things that are fundamentally susceptibility in terms of autism or how the brain works differently. So as we've done that, just to acknowledge our wonderful community that's come together in terms of this, we've had people from around the country, and I'll show you some numbers in just a little bit, who have been incredibly generous in terms of making this a community effort to understand this better. And so these are just a few of the wonderful people who have made Spark what it is. So I'm going to let you see some insight into what it is that we actually do at Spark Central as we're going through and looking at this information. So here's an example, and shown across the top is what we call a reference, or the most common genetic alphabet code that we see for a particular segment of the DNA. And it's filled with the alphabet letters A, U, G, and C. Those are the 4 alphabet letters that our genetic information is coded in. You can see, and I've shown here in the circles that are in yellow, a difference between 2 individuals. In one case, it's a C, in one case, it's a U. And in particular, that alphabet code is used to make what we call amino acids, or building blocks of proteins. And proteins are what make up the way your body is built. They're the structure, they're the function in terms of how we can do what we do, including how our brain works. In this particular case, this change of a C to U, in fact, doesn't change that protein, doesn't change that amino acid of glycine. And so in those particular cases, we assume those are what we call silent. They don't have an effect in terms of our body's function. On the other hand, and I'm going to show you a couple other examples, there may be circumstances in which a similar sort of change, just one single alphabet letter, in this case a G to a C, changes one of those amino acids. It changes an alanine to a proline. Just seeing this change alone, I don't actually know whether or not that will functionally affect how that protein works. It could be that it changes the way the protein works. It could be that it has no effect on that protein. That's not something that I can necessarily determine just by looking at this information. On the other hand, sometimes there are other changes, in this case an A to a U, which changes this amino acid from a lysine to a signal that tells the cell, stop making this protein. So it introduces what we call a stop, or being able to prematurely terminate that protein. In many cases, we think stopping short of making the full protein is, for that protein, not what it was meant to do. And so in those cases, it's easier for us to interpret what that means. However, the vast majority of genetic differences that we see that we consider as being related to autism are in the former class, what we call those missense variants, or those amino acid substitutions. And I'm going to get a little bit more into that in a second. So as we're doing this, within Spark, it's extremely expensive still for us to sequence the entire 3 billion base pairs of every single person in Spark. So we've taken a shortcut. We don't think it's actually a bad shortcut to take, but this is what your genome looks like. It's quite complicated, as you can see, and hard for us to understand what it all means. And so we've taken a shortcut, which is to look at the golds or the yellow colored regions that I've shown here. These are the portions of the genes which actually make the proteins. We call them the exons. And when we put all of these exons together, we call it the exome. And if we aggregate all of this, it accounts for a little less than 2% of the information that you have genetically, but it accounts for, we think, about 85% of the genetic changes that, at least right now, we know how to interpret. And so we've taken this as a first pass for many people in Spark because it's the information we know what it means, and we know what it means in terms of brain function. In addition to those changes, which are just single letters, there are also normal variations in individuals, which actually look at not just one letter, not just one protein or one gene, but can look at a sort of aggregated neighborhood, is what I've called it here. And so I'm showing here a region on chromosome 16, and you may be able to see a little golden block, a box around an area. Down below, I've blown that up so that you can see what's involved in that. That's one region, one neighborhood that has 29 different genes. And for some people, instead of having 2 normal copies of that neighborhood of 29 genes, they may have only 1 copy. So they have a deletion or missing region of chromosome 16p11. Other individuals actually have that duplicated. They have an extra bit of information, and so they have 3 copies of that region. And we know, and have known for a long time, that this region, what we call deletion or duplication of this region, is the most common genetic reason that we see for autism that we currently understand. And so this is one of the things, one of the results or types of results that we see within Spark. I'm going to use a terminology called copy number variation, or CNV, to represent these neighborhoods that sometimes can be either missing or extra. Then you'll see in a second that these are quite commonly a cause of autism. So as we've been doing this, we have intentionally set the Spark study up to be able to better understand genetic causes when we have parents available. And so for many of you, you'll realize that we asked you to send in saliva samples for the individual with autism, and when it was also possible to include the mother and the father. The reason for doing this is by being able to do a direct comparison to both biological parents and the individual with autism is that we can very quickly recognize the things that are new in the individual with autism, those so-called de novo events that I told you about before. Because there is an overwhelming amount of information we see genetically, we need something to give us clues into where we should be looking. And so it is surely only the first sort of generation of being able to do this, but that first pass is oftentimes very relatively easier to do by looking for those new genetic events. And so we thank all of the moms and dads out there who have made their contributions as well. So as we think about this, and I'm showing you just a typical, we think of it as a family. Many of the families that we've had in Spark have included, as I said, a mother, a father, an individual with autism, and for some of you, you've been kind enough to also include multiple of your children or multiple siblings. In some cases, we can see that we have a new genetic change in the individual with autism. In other cases, we can see that there are things that are passed down from the mother or the father. One question that parents frequently ask me is, if my child has one of these new genetic changes, what happened? What caused it? Was there something that I did or didn't do that made this so? In point of fact, as we're doing these analyses, the samples that we have from you are from saliva. And I think you know this because you've given us saliva samples in the cup. What we are looking at is necessarily a portion of the body, but is not entirely representative of the body. And in particular, when we do that, I can't tell whether or not a de novo genetic change came from a mother or a father. All I know is that it was new in the child. It theoretically could have been present in the egg or the sperm at the time of conception, or, as I'll show you about in a second, it may have happened after the point of fertilization. And again, because we're not checking egg cells or checking sperm cells, we can't really know when that happened. But as we're doing this, as I'll show you in a second, some of these things may have been very old, rather, in terms of egg and sperm, and have a small chance that they could happen again. That is, that if it were present in the sperm, as an example, it could be that there are other sperm cells affected, and there could be, even though it's new, something that has about a 1% chance of happening again. In certain circumstances, this has happened after the point of fertilization. And so what I'm trying to show you here, all the way on the left, you may be able to see an egg cell and a sperm cell coming together. After they unite, we make the zygote, or a very early embryo, and then those cells begin to grow and divide and eventually develop into a human being. It is possible that that genetic change, again, was not present in the egg or the sperm, but happened at some point later, what I'm showing you in green. And when that happens, the individual is actually made up of two different populations of cells. And we call that mosaic, just like a mosaic tile you might see. And sometimes we can actually see two different populations of cells genetically. The reason I mention this is because those individuals, there is a different risk for the families in terms of this happening again. That risk is lower, is actually not going to happen again for parents when we know that that mutation is mosaic. And there's more and more evidence to suggest that some cases in autism with de novo mutations are due to that mechanism. In addition to those new genetic changes, though, probably a significant fraction, and in fact a large portion that we don't understand yet, also come from inherited genetic variations. And this gets complicated for us for reasons I'll describe in a second. So as we think about this, we can think about genetic factors that can be of different weight. Even though we have all things that I might call genetic and might be in A's, T's, C's, and G's, some of them have big impact and some of them have smaller impact. So I'm showing you on the left, this large blue square is meant to represent certain genes or certain genetic variants that have a very big, powerful impact. And I'm showing you on the second from the right, some smaller squares, orange squares. Then they're smaller because it's meant to represent that they have a smaller impact. And in fact, these also have to do with numbers. So there may be individuals who have just one big blue square, one big blue gene, if you will, where that is what we think the major contributor to autism. Other individuals in which it may be a combination of multiple, what we think of as more common genetic variants, each of which has a relatively small effect, but in the aggregate, in the sum, putting them all together may have a higher risk of autism. Now, one of the questions folks have been asking me in terms of Spark is, are there certain individuals who are more likely to get a genetic result returned to them? And so I wanted to explain this a little bit further. There are individuals, as I talked about the heterogeneity, there are individuals who seem to have certain types of challenges. So as an example, there are some individuals in Spark who have problems in terms of intellectual disability. There are certain individuals who are older, who still have not yet learned how to talk, they're nonverbal, and some who may also have certain medical problems, be they epilepsy or certain types of things that they were born with, congenital heart disease or other problems. Those individuals are actually the group where we are today, at least, more likely to identify something that we understand well enough to return it to you. And so for those individuals, they are more likely to have one of these sort of more powerful genes that we recognize and that we currently, I'll get to in a second, but understand to be able to explain to you and to be certain that this is actually the cause of their autism. Over time, what we anticipate is that we are going to, with larger and larger numbers through the research studies of Spark, be able to identify genes that have smaller effects. So not just the big blue squares, but also some of those yellow medium squares and even the smaller orange squares. With that, though, it's basically a matter of how large our sample is to be able to recognize these things. So in other words, we need to have more people that we can study all together to be able to recognize genes with smaller effect. And so as I'll be showing you about, we're still in the relatively early days of Spark in terms of putting the numbers together so that we are more likely to see genes of large effect and eventually be able to get to genes with smaller and smaller effect size. So as we're doing this, I wanted to give you a flavor of what we've been learning from Spark itself. And I think one of the things that we certainly celebrate along with our 27 clinical sites around the country is just how incredible the community has been about coming together. As of today, we have over 160,000 individuals participating in Spark across the country. That accounts for about 63,000 individuals with autism, and this comes with individuals who have participated in multiple different ways, including most in the genetic studies. To give you a sense of the time frame and what we actually go through and why it takes a while to hear from us, is because there are multiple steps within this process. And as we're doing this not just with every person as they come through, but we're putting things together in groups so that we can do things more efficiently and more robustly. The first thing that you all are very used to, you know about, is being able to provide us with the saliva sample. So the first thing we have to do is be able to extract the DNA from that. I will tell you that although it's usually successful, there are cases in which we are unable to get enough DNA from the samples you provide, and we have oftentimes sent out requests to ask you to send in a new sample. It's real important if you are interested in being able to be part of the genetic studies and perhaps get results back, to send in that replacement sample, because without that, we can't get beyond step one. So again, thank you for those who have supplied the sample. When we do have adequate sample, we go on to the sequencing. That is, in most cases, exome sequencing to be able to generate the information that we need to. We generally do those in very large batches, and it can come down to usually once or twice per year that we're doing these very large batches. We can't predict when that will be, so we like to try and get everyone through and make sure we've got good, high-quality DNA, and then we put everyone together to do it in a robust way. After we get the sequence data, and it takes us, again, just so you know, time frames about this, it takes us, because these batches are so large, oftentimes 3 or 4 months to be able to get all of that sequencing done, and then we come back with one big batch of data to do the analysis. There is a team at all 27 of the clinical sites around the country. We have members who are part of what we call the genetics working group to be able to analyze the data. We have a lot of brilliant people that are putting their heads together to make sense of this in multiple different dimensions. And so they all participate in step 3 of the analysis. After that, we like to double check everything we do. We want to make sure that we're giving you the right information. And so with that, we also go back and do an independent second look to make sure the information we think is relevant to autism for you is in fact correct. So we have new eyes look at that and make sure that's correct. With that, we go back to a new sample from the DNA and again, do an independent confirmation. So again, one of the reasons why it's so important that we have a good DNA sample and have enough of it is because we need to do that confirmation. I will say there are some of the families we've actually requested a second sample where we are stuck on step 4. That is, we would like to be able to do the confirmation, but we do not have sufficient DNA from your sample to be able to do that. And so we're waiting to go on to the next step. And if you got one of those requests again, please do send that in if it's something that you think you would like that information. And finally, if at first we don't find the answer, we go at it again and again. And as I'll show you in a second, we certainly understand more and more each year that we go back. And so as we'll get into, this is a continuing process. To give you an update, in this first pilot that I'll tell you about, we had 11% of families that we looked at that we did have an answer that was available for return. And we anticipate that in the future, that number will continue to increase. So 10% or more is what I'm expecting. So far across Spark, here's where we stand. There are 500 families where we have completely finished all of those steps 1 through 5 that I've talked about in terms of being able to do the analysis and going into the process of returning that information. For those of you in that first 500, if we didn't see something that was ready for return, we sent you an email letting you have a status update that we had sequenced and analyzed everything, but we don't recognize anything at this point that's returnable for you. There are 9,000 additional families that are in the process of stage 3 right now, where they're undergoing analysis, and we're starting to do stage 4, some of the confirmations. And then there are additional 6,000 families right now that are in the process of stage 2. So they're getting ready for the sequencing step. And then there are more families that are coming in that are earlier in that process. Within this, our first pilot study was of approximately 500 families. And of the 11% that we returned, there were several different types of genetic changes that we saw. And this is showing you a breakdown of this. About half of them, as I said, a little more than half of them actually, were de novo genetic changes or new in the individual with autism. But you'll also see in the upper left-hand corner that there were a fair number of these that were inherited from parents. When they were inherited, these particular types of genetic changes are not always associated with autism. They may increase the risk of autism, but it is not a one-to-one equivalent of you have this genetic change, therefore you must have autism or you must have something else. They can just be normal differences that we see within the community. So as we're telling you about these things, like I said, some of these have different manifestations and different meaning in those families. So I wanted to show you some of the sort of very detailed analyses that we've done. So I'm showing you here that what we did is not just looking at our 460 or 500 families that we looked in with Spark, but we also looked across the published literature for other cases of autism that were part of other studies. And we put all of this together, realizing that there's power in numbers. And we wanted to see if we could start to see any patterns by putting everyone together. What I'm showing you along the top line is that we identified many genes. So those are the names of the genes shown along the top. So CHD8, SCN2A, different genes that we know are related to autism. Shown in that middle bar are genes that we knew, some we had already known were related to autism. Those are the ones on the left. And then some that we didn't yet know were autism. And those tend to be ones that were on the right. That bottom, or that middle row rather, that's showing you the different shades of red are from a statistical point of view, telling you how strong our signal was coming from our individuals in Spark compared to the rest of the, combined with the rest of the community. So the genes that were on the left, we were very, very confident of. We knew for sure that those were related to autism in those individuals and that across others would be related to autism. In the middle, with that kind of lighter red area, those are genes where we felt confident, but it wasn't quite as confident. And then the ones on the right, we didn't know. These were new genes for us. The ones that are shaded in that greenish blue were genes that were, for the most part, not known to be associated with autism. And so we were more, guessing a little bit. We had some hunches, we had some ideas, but we weren't confident about this information. And that confidence to me is real important because we want to make sure that we're giving you information that we can be really confident, you can be really confident in. And sometimes that means we need to see it over and over again, not just in you, but also in other families like you. And with that, the whole point in Spark is that as we look at more and more individuals, we will get more confidence and be able to return more information to you. So as we're doing this, the next wave, as I said, that'll have about 9,000 additional families, will take both the first 500, include another 9,000. We'll also include anyone else in the community outside of Spark, and we will look again. So it'll be like turning the wheel one more time and seeing if there are things that in our first group, maybe we didn't recognize because we had a pretty modest number of individuals, 500 individuals. But now as we think about how much larger the community is, as an example, 9,000 you'll realize will be then more than twice as many as the entire world had together for autism before that. I mean, just think about that, how much more certain we can be by adding those numbers from Spark. So the idea is that over time, even if we didn't have an answer in year one, maybe we will see something and recognize something in year two, because we will be confident having seen it not just once, but twice or 3 or 20 times, but we will have statistically been able to be sure that what we're giving you is the right information. To give you an example of this, one of the genes, and if you went back a couple slides, you would see these two genes across the top. One of the genes we saw was a gene called BRSK2. Within Spark, we realized that this was a new genetic change, and everyone that we saw shared some similarities. They happened to all be male, but they all had problems in terms of expressive language. Either they were nonverbal or minimally verbal and had intellectual disabilities. They seemed to cluster together. In fact, at the time, this was a brand new gene that had not been associated with autism, had not been associated with any sort of intellectual disability. And in the process of doing this, in fact, for the first time, a report came out in the literature of this gene being associated with what we had seen in Spark. So we suddenly became much more confident of this information. In a similar way, but different, we saw a second gene, ITSN1, again, that had never been seen before in terms of autism. In this case, this was a different profile. Individuals had autism for sure, but they tended not to have language problems, not to have intellectual disability. They seemed to be just different. They were clustering in a different way. And in this case, we saw that some of our genetic changes were new, but many of them were inherited or passed down within families. And this might represent, just as an example, the top one of those big blue squares. Maybe this bottom one is more like a medium-sized orange square. But they may be different both in terms of their profiles as well as in terms of the impact they have on the brain and behavior. So in terms of this, it's been great to get to know some of the families. I've had the pleasure of literally talking with many of the families and talking them through what their results mean. So I wanted to share that information back with you. So one of the things that I've heard from the families is just what a relief it is to have an answer, to be able to answer this question of why. And for some families that I've had the pleasure of speaking with, they've been wondering about this for a year or two. I have to tell you, there have been some families that have been thinking about this for more than 20 or 30 years. And so it's been incredible for them just to have the end of one journey they've been on, trying to find an answer and being able to answer that. With this, there have been some families who have been wondering about the question of, will this happen again if I have another child? Or in some cases, it's a sibling of someone with autism that's been wondering for themselves whether or not they might be at increased risk. And to clarify that has been helpful. And as they've gone through that journey, that's not to say that they don't, are not incredibly grateful for the family members they have with autism and how special that relationship is, but they do know that there are certain things that come along with that, with having autism in those individuals. In addition, one of the things that's been helpful is trying to get some information about prognosis, to have some idea about what the future might hold. So I'll give you one example. One of the families that I spoke with, their child was still little, but he was nonverbal, and they were wondering to themselves, they were spending a lot of energy on speech therapy and trying to think about what the best way of being able to help him communicate was, and they just didn't know, were they beating their heads against the wall for nothing, or was this eventually going to work? And based on his genetic form of autism, we did know that early on, speech was going to be a huge challenge, but we also knew what kids looked like when they were 10 or 15 or 20. We knew what things were like down the road, and we knew that all those kiddos were in fact verbal, that it does take a while for speech to come in, but not to give up, and also to be able to give some advice in terms of strategies that might be more productive, and they found that quite helpful. In addition to that, one of the things that's been enormously powerful is just being able to meet other families like them. So being able to realize that they're not alone, be able to join a Facebook group for other families that share similar challenges, to realize that across this complex group of individuals with autism, there might be a certain subset of them that in fact have very, very similar challenges to what they've seen in their family, and to come up with some very practical tips about how to deal with that and what to expect in the future. And for some of those families, they've actually come together, physically come together and met with each other, and just being able to share that experience, I think, has been uplifting for them as they've done that. Although it's not the case yet, what we do hope is that for number 5, we will get to the point that we will have more focused ways of being able to help support individuals that is based on these individual genes, either because we know about the prognosis, we know certain challenges that may come up in the future, or because we understand the molecules in the brain and how they're working and have very much more targeted approaches for them. Within all of this, again, I think the vast majority of families that I've dealt with may have had some combination of ways in which this is beneficial. It's different for different people and at different stages in their or their child's life, but I think everyone so far has found something that's been useful out of this experience. After we find the information, I want to emphasize that we've thought about what next steps might be helpful for families. So it's not just giving the diagnosis, but it's also thinking about supporting them in the next chapter of their journey. And sometimes this can be unexpected, not necessarily knowing what the next step is. So we've tried to make this roadmap. With this, we've had a program for many years called Simon's VIP that Vincent mentioned earlier. It's a community that we have around individual specific genes that are related to autism, and we have many members of the community, some of whom have come through Spark, many more of whom, at least right now, have come because they've gotten a genetic diagnosis from their doctor, their geneticist, their neurologist, and they've now wanted to be able to network and learn from other individuals with the same condition. So we've set this up to be online, so that would allow families from around the world to be able to come together. We've created a community so that they can share information with each other. We set this up so that it can be de-identified. No one has to know that it's coming from any one person or be able to identify who they are, but be able to collectively share this information across the community. We set this up so it's a partnership with researchers to be able to make it easy for people to do research in that condition. Again, all of the information is de-identified, but we make it so that families only have to contribute their data once, and many, many researchers can all use the same data without having to bug you multiple times. In addition, we've created what we call a biospecimen repository. In other words, samples, oftentimes of blood samples or cells that researchers can use to, again, be able to understand how the brain is working differently, and important, be able to, in the long term, we hope, support individuals better and perhaps come up with new treatments in the future. So as we've done this and as we've given the information back, we realize that Spark cannot and should not be doing everything in terms of Simon's VIP. We realize that this information is something that we hope that you'll share with your other providers, your other child's providers, whether that might be your doctor. In some cases, it might be therapists. In other cases, it might be teachers. It might be a local genetic counselor or geneticist that helps you think about what this information means within the context of your child or your family member. And again, because many of these conditions are rare and your individual doctors or providers may not ever have known someone with the same condition, we encourage them to use the information we have at Simon's VIP. And that website, simonsvipconnect.org, has things like WebExes that I and others have done to provide a lot of detailed information that we hope is useful in terms of medical management as well as educational strategies. So that's helpful for about 11% of you. And the other 89% of you have been wondering, well, what happens to me? So one of the things I want to emphasize is that you may have gotten an email, again, if you were in that first group, that says, at this time, we don't have a genetic result for you. That's not to say that there is nothing genetic. It's not to say that we figured everything out, because we certainly haven't. And we certainly haven't given up either. We continue to work on this. And it may be that even though we don't have an answer for you now, we may have an answer for you in the future. In the meantime, though, if there is something urgent for which you need a genetic answer immediately, either there's an issue of a pregnancy in the family or something else, some other medical issue that's emergent, I do urge you to think about using a local genetic doctor, a genetic counselor, a neurologist, someone else who can help you get the information very quickly if you need it on a shorter time frame, because again, as you can see, this goes I think it moves at a pace that may not be quite as fast as you might want it to be. So as we go through this, you will also be able to see, besides an email that's sent to you, your dashboard. So your dashboard on Spark gives you an up-to-date status in terms of things like where your saliva sample is in this process of sequencing, those sort of stops that I said along the way, as well as various different questionnaires that we've asked you to complete. And if for some reason you forget where you are in that process, feel free to log into your dashboard and you can get an up-to-date assessment of where your sample is within this process. Some people also wonder, they say, well, goodness, you've sequenced our entire genome. What else have you found out about us? So I want to be clear that we're not trying to find out about everything about you. We really are focused in terms of looking for autism. We've got our hands full in terms of being able to do that, and we're trying to stay true to that Spark mission. So that's really where we're putting our time and energy in. If we do happen to see something that we think is related to some immediate medical concern for someone in your family, either something like a cancer risk or something like a sudden heart problem, something that might suddenly cause the heart to stop, if we see that and recognize that, for those of you who have requested that information, we will have the opportunity to get that back to you. But again, do not rely on Spark to give you that information. If there is a family history of cancer or some heart disease, certainly see your doctors about that individually. So in summary, we've come a long way. We certainly know much more about the causes of autism today than we did a decade ago, but we certainly have a long way still to go. Within this, again, although I've talked a lot about genetics today, I want to be clear, it's not all about genetics. Genetics are an important way to understand biology of the brain and behavior, but it's not that everyone's cause either is genetic entirely or even in part. As we're doing this, we do hope that we will, again, be able to give information to certain individuals right away, but that we continue to do this iteratively over and over again. And as we're doing this, we're also building a community of individuals beyond when they get a genetic answer to be able to come up with more answers for the future. It's been my real pleasure to be able to speak with you today, and as well to continue working with you through all the things that Spark does in the future.
Okay, thank you so much, Wendy. We did get a number of questions. We'll try to work through as many as we can. So it's just going to take me a second to sort through some of these. Okay. So Can you just explain, I think you might have missed this in one of your discussions. So when you register for Spark, how many siblings can enroll?
Okay.
Yeah, so you can just.
Yeah, so in terms of Spark, traditionally the way we're doing this is that if there are multiple siblings with autism, then certainly all the siblings with autism are invited to enroll. Also, if there's an extended family that has other individuals with autism, certainly we would welcome all of those extended family members as well. In general, in terms of the number of siblings beyond those with autism, we'd like to have, for families who can, one other sibling without autism to be included within Spark. And by doing so, the reason some people might wonder why we do that. So one of the reasons is because in some cases, we like to be able to understand what might be similar, either exposures within the family. Again, there are some things that are shared within families besides the genes, as well as when we start to look at some genetic variations, we're not really sure what they mean. And so being able to look at those variations in multiple family members and see whether or not they travel with autism consistently is helpful in terms of us understanding whether it's cause and effect or simply coincidental.
Can you talk a little bit about the process of return of results? Someone was asking if participants receive genetic counseling.
Oh, very good question. Thanks for asking that. I didn't address that at all. So for individuals, when you first register, when you first consent, you don't have to participate in the genetics, number one. You don't have to get results back, but for people who say they would like to, and I will say that for the vast majority of individuals who submit saliva samples, they do want to get the results back. You check that at the time of consent. As we have the results for return, we ask you again, do you still want to have that result? Again, the vast majority of people had said yes, we would still like to have that information. We give you a number of options in terms of who returns that result to you. Again, we have independently confirmed this to make sure it's correct, and we make sure that it is concordant with what is both most convenient and most helpful to you. So what I mean by that is some of you have said, you know what, I have a genetic counselor, I have a genetic doctor, I have a neurologist, I have a pediatrician that we work with very, very closely, and it would be great to have them return that information to us. We know them, we trust them. And so in those cases, we fax a copy of that report to that doctor, to that provider, and we have you talk with them and be able to understand that result through them. We include some information that is both a copy of the genetic test report as well as what we call a gene brochure. So information that we at Spark Central have curated by combing through the literature, knowing everything about the gene and what it's associated with, and putting it together in one consolidated place that's helpful, we hope, for families as well as for the providers that work with them. So you get a copy of that, both the genetic test report as well as the gene brochure, and your doctor does as well. Other individuals, they may not have a provider that understands this, that feels comfortable with the genetics. And so at Spark Central, we have paid for genetic counselors who can be conveniently available to families free of charge. So this is all paid for by Spark, to be able to explain the results to you on the phone and to also, as I said, provide you with the genetic test report as well as the gene brochure. In some cases, I have been that geneticist talking with the families over the phone. I won't be the only one in terms of returning this information, and we think that acts as a good bridge in terms of being able to immediately hear about the result and understand what it means, but a bridge to the individual's own doctors, to their own therapists, to their own providers, and so we certainly want that information to be shared when you have that report with your doctors, with your pediatrician, with your neurologist, with your internist, so that they can think about helping you to start the next chapter of your journey and using this new information to get to where you need to go.
Okay. Someone is asking about if a genetic change is de novo for a given individual, is it possible for it to be inherited in future offspring?
Oh, that's a great question. So you guys are real sharp. So even though this is de novo in the individual with autism, for the cases that we've seen so far, these are things that can be passed down to the next generation in a 50-50 sort of way. So let me try and say that again. So even though this is new in the in the person with autism, it's a new genetic event for them, they have two copies of that gene, one with the change, one without the change. And when they go on to have their children, it's a 50-50 chance which one of the copies of the gene will get passed down, but it could get passed down to the next generation. Now, whether or not it's associated with autism in that next generation is an individual thing that that person should talk about with their team, their genetic team especially, because it's not always 100% that that gene will cause autism in the next generation.
Can you talk a little bit about how genetic findings related to autism can contribute to the understanding of other conditions such as ADHD, anxiety?
Oh, great question. Boy, you guys are on fire today. So one of the things that I was saying at the beginning is that not everyone colors in the lines with these genes. What I mean by that is that although they can be associated with autism, for some people they can be associated with ADHD. For some people they could be associated with anxiety. There might be other behavioral differences or other differences with the way the brain or other parts of the body work. At the same time that Spark is focused on autism, there are many other groups around the world that are focusing on other conditions. There are folks that are focusing, for instance, on ADHD, or folks that are focusing on seizures, or folks that are focusing on intellectual disabilities. One of the things that we can do is all come together and be able to compare notes. And in part, that's what Simon's VIP is set up to be able to do. It takes communities and builds communities based on the gene first, and then secondarily thinking about what symptoms you had or what manifestations you have that are related to the gene. So in that way, once you do get one of these genetic diagnoses, we can understand better the full spectrum of the different challenges associated with it. And so between these two dimensions, we hope that we'll be able to make inroads. But yes, these are complex brain differences, and they're not always just monolithic in terms of their manifestations.
Okay. So I've seen some questions about people that have participated but not sent in samples yet. So, you know, how old would a kit have to be too old?
Great question. So for those of you who may have a kit, it may be buried in the bottom of your cabinet or dresser drawer or something. The kits are actually incredibly robust and last a long time. So for most of you, it's actually still going to be okay to be able to spit in that saliva sample. The postage is still good in terms of sending things in. For sure, if you've received your kit within the last year, there is absolutely no problem with it. And I have to say, I've used kits that have even been 3 or 4 years old and used them quite effectively. So if it's relatively simple and straightforward to be able to get a saliva sample and you've got that kit on hand, I would say go ahead and send it in. If it's something that's a bigger struggle and you're worried about whether or not you've passed your expiration date, send us a sample in. You can contact us through the Spark website, and we'll make sure that we've got a new kit if your kit is expired.
So I've seen some questions about different kinds of testing. So can you talk a little bit about maybe kind of the trajectory? So there was at one point only one kind of testing, and now we're moving forward. So someone had mentioned karyotype. So can you kind of walk people through the different types?
Yeah, so there are, I think of them as macro or big genetic changes and micro or small genetic changes. And so a karyotype is a chromosome difference. It's sort of a big difference. We see a big amount of genetic information that's different in those people. That's something like Down syndrome as an example. We had been able to get more resolution for that. So the analogy I sometimes use is a karyotype or a chromosome difference would be the equivalent of missing Australia if you were to look at the globe. It would be a big difference you could see. There are some things that are portions of chromosomes, so they're smaller units. That might be something, for instance, at the state level, where you might be missing New Jersey in that particular case. You can even get down smaller, so you can get down to the level of a zip code. Those might be, you know, a couple genes that are different. You can even get down to the level of a single street address, and that's when you get to the level of a single letter that's different. Now, the thing that's sometimes confusing is size matters, so that's important, but it's not just size. It's like real estate, it's location, location, location. And so it's not just the size of the genetic difference, but it's the location of the genetic difference. And sometimes, even though it's just one letter that's different, if it happens to be just right in the middle of a very important gene for brain function, or if it happens to be a particular type of change like that stop sign I showed you, can have a big impact even though it's a small genetic change. The techniques that we're using right now have the ability to see all those different types of change almost. Remember I told you we were sequencing about 2% of the genetic information for most people. We may have opportunities to go and do it in even more detail, getting up to even closer to 100%. What we're trying to do is get as much information back for the community as well as for families as we can. And we'll do this as sort of opportunities arise as sequencing costs change.
Okay. So we are unfortunately out of time here. We did get a number of questions. If you'd like, you can send any additional questions to info@sparkforautism.org, and we will work to get you an answer.